scientists believe that peptides can be developed as effective competitors against harmful protein-protein interactions within cells. However, peptides found in nature contain certain limitations, such as their rapid degradation and their inability to cross cell membranes, that prevent their application as effective competitors. Samuel Gelman and his team have found that alpha-beta peptides developed in labs could be a solution to this problem. When tested in the past, alpha-beta peptides show decreased susceptibility to degradation and have mimicked the beneficial properties of natural peptides when designed properly. Additionally, their helical backbone modification and their hydrocarbon cross-linking, which acts as a stapler, should make them stable enough to be used in real-life scenarios. Ultimately, Gelman and his lab team decided to run tests on these alpha-beta peptides to ensure that they don't degrade rapidly, can bind to proteins, are stable, can cross cell membranes, and, in the end, successfully compete against harmful protein-protein interactions found within cells.
The first test performed was the surface plasmon resonance, or SPR test. SPR was used to evaluate the affinity of alpha-beta peptides to proteins. In SPR, the lower the IC50 numbers indicated a stronger affinity between the individual peptides and proteins. There were 6 peptides tested with 3 different proteins. 5 trials were performed to ensure the results were genuine. From the results showed in the table, we can see that the alpha-beta-1 peptide has the highest affinities across the board with all 3 proteins. Also, we can see that the alpha-beta-1 peptide had the longest half-life.
Circular dichroism is the method used to test for stability of hydrocarbon crosslinks. Circular dichroism is the difference of left-handed circularly polarized light and right-handed circularly polarized light, and occurs when a molecule contains one or more chiral chromophore. The cytochrome C release is a method used to test the ability of peptides to induce cytochrome C release from mitochondria, which is the powerhouse of the cell. Results indicate if peptides can cross membranes. In this specific experiment, the peptide showed that alpha-beta peptides were able to cross the cell membrane in a negative MEF environment.
The last test Gelman ran assessed whether the alpha-beta-1 peptide had the ability to kill cells. This could be beneficial in nature, as the peptide can be used to signal the death of cancerous and dangerous cells. In the test, various cells were exposed to the alpha-beta-1 and alpha-1 peptides for a set period of time. The results, seen in the figure, show that the alpha-beta-1 peptide had the ability to kill cells, with every trial resulting in a success rate of over 60%. In conclusion, it was found that the alpha-beta-1 peptide can be used as effective competitors against dangerous protein-protein interactions within the cells. This is due to the peptide's characteristics found by the tests explained earlier, like its affinity to proteins, minimal degradation, ability to cross membranes, stable, and ability to kill cells. This quality means that the alpha-beta-1 peptide may be used for the development of therapeutic agents in the near future.